So in this lesson, you're going to look at the function of collecting duct. So the function of collecting duct is osmoregulation. Osmoregulation. Okay. So what do you mean by osmoregulation? Osmoregulation means the control of water potential. Regulate means to control. Control what? Osmo means water potential. Okay. Water potential of what? Of the blood and tissue fluid and also the limb. Okay. The blood, the tissue fluid, the limb. These are all the internal environment. Okay. So how does it control the water potential? By controlling two things. Firstly, water content. Because you know that water potential is affected by two things, right? Water potential is affected by how much water you have and how much solute you have. Okay, so therefore, body will try to control the water content as well as the solute. Okay, so by controlling the water content and or the concentration of ions, particularly sodium ions. Why sodium ions? Because every day we eat, we take salt, right? Every day we eat, we take salt in our food, isn't it? So there'll be a lot of salt and water going in. So the main solute that we want to control is sodium ions. Okay. So this regulation, okay, is actually regulated by antidiuretic hormone. So antidiuretic hormone official short form is ADH. Okay, so ADH is the official short form, antidiuretic hormone. So what do you mean by antidiuretic? So let's look at the meaning of the word diuresis. Now, diuresis, cis is a process. Any word that ends with a cis is a process. Okay, diuresis means production of dilute urine. So that's why I put pink color. Diuresis is a process where we produce dilute urine. Dilute urine. Okay. So when our body secrete antidiuretic hormone, what kind of urine we are trying to produce? Anti means opposite of. If it's opposite of dilute urine, meaning concentrated urine, right? So that means to say that when we secrete antidiuretic hormone, antidiuresis means we want to produce a concentrated urine. Okay. So therefore, ADH causes the production of concentrated urine. So let's study the regulation of our the osmoregulation by ADH. So we're going to study the osmoregulation by ADH. It is based on the stimulus response model because you know that hormone, right? Hormone is a signal molecule that's involved in the transmission in the entire stimulus response model, right? It's part of the stimulus response model and which is used for homeostasis. So the homeostasis here is osmoregulation, regulating water potential of the body. Okay, so therefore, whenever you want to study, whenever you want to study and to explain the homeostasis process, in this case here, osmoregulation, it's best to use the stimulus response model as the base for you to write your answer. Then you have got a lot of points to write. Okay, so it's a guideline for you. So since this is the base for the osmoregulation, we're going to look at the stimulus response model again. Okay, so stimulus response model is made of stimulus, receptor, transmission to coordinator or regulator or control center, transmission to effector, and then response. So the definition, stimulus is changes from the set point. Receptor is a specialized group of cells which can detect the stimulus, convert it into a signal to be transmitted. And the transmission in our body is always by nervous system or endocrine system or both. So nervous system is transmission of the signal in the form of nerve impulse transmission along the neuron, which is the nerve cell. Endocrine system is they secrete hormone as a signal molecule transmitted in the bloodstream from one part of body to the other. Means from receptor to coordinator, from coordinator to effector. Okay. Effector can be muscles, glands, organs that will produce the response, and response is an action produced as a result of stimulus in order to restore back the set point. So this is a stimulus response model. So applying this to understand osmoregulation here by ADH. So the stimulus here must be a decrease in water potential of the blood, isn't it? Okay. So decrease below the set point. So it must be changes away from the set point. Okay. So there'll be receptor, which is osmoreceptor in hypothalamus. So osmoreceptors are specialized cells that can detect the change of the water potential of the blood. These osmoreceptors are these cells here. Okay, these two cells here labeled here, and they're located in hypothalamus. So this is hypothalamus. Exact location, if this is the head of the person, this is the brain, right? This is the front part of the brain. So we call this forebrain. So hypothalamus is located at the base of the forebrain. So at the base here, this region here, okay, which is above this P-shaped gland. So this P-shaped gland is right above our ear. Right above our ear, inside there is the P-shaped gland. So hypothalamus is located at the base of our forebrain and above the P-shaped gland called pituitary gland. Okay. So hypothalamus located in the base of forebrain above pituitary gland. Okay. So what happened after the receptor is being stimulated by the stimulus? So this receptor will then send a signal. So this is called transmission. And this signal is sent by the nerve cell. So nerve cell is also called the neuron. Okay, in the form of electrical impulse. Why you please write your notes, huh? Okay, it's not important, I will not type there, okay? So once they detect that the blood water potential has dropped, they are being stimulated, and they will send electrical impulse along the nerve cell, because this receptor is also a nerve cell. Huh? This receptor is a cell, is also a nerve cell. So it will send electrical impulse along the nerve cell all the way. So it goes down from hypothalamus to the posterior pituitary gland. Can you see this pituitary gland is divided into two parts? Okay, can you see this line here? This line here. So pituitary gland is divided into two parts. Its anterior pituitary gland is the one facing the front. Okay, the one facing the front, which is here. So this part is called anterior, isn't it? Okay, it's in front. Okay, and then you've got the posterior pituitary gland, which is this one here. It's called posterior because it's facing the back. So this person's back is here. Okay, facing the back. Okay, so anterior, posterior pituitary gland. So the electrical impulse will go all the way to the posterior pituitary gland and will arrive to the end of the neuron. So this is the end of the neuron, right? The end of the neuron will secrete a hormone called ADH. So ADH will be released by this nerve cell at the end of the nerve cell here. Okay, so this secretion, so our coordinator is posterior pituitary gland, because this nerve impulse go into the posterior pituitary gland. And then it will lead to another signal to be released. So this signal is actually the ADH. So it's a hormone. Okay. And because it's released by the nerve cell, it's called neurosecretion. Neuro means the nerve cell will secrete the hormone. Okay. So specialized nerve cell will secrete the hormone. So this is called neurosecretion of ADH. So this green color arrow shows you that ADH is secreted into the surrounding tissue fluid. Right? Surrounding the cell is always tissue fluid, right? So hormone is secreted to the tissue fluid, and from tissue fluid, it will go into the bloodstream, right? This is capillary. So it go into the bloodstream. So then ADH is transported in the bloodstream. Okay? So next, what happened next is the blood will transport the hormone all throughout the body, but only the target cells. will be activated by the hormone, right? Okay, because the target cells has got receptor. Okay, target cells got receptor. So who are the target cells? The cells of the DCT and collecting duct. So these two are the effector. Okay. So what happens when ADH binds to the receptor of DCT and collecting duct cell? It will cause the wall of the cell to become water permeable, so increase in water permeability and increase in urea permeability. It means the membrane of the cell becomes permeable to water and urea. So we're going to look at this one in detail afterwards. Okay, over here. So this diagram shows you on the right side is the capillary around the DCT and collecting duct. Okay, this is a capillary carrying the ADH all the way from pituitary gland and arrive to the kidney already. Then this is the endothelial cell of capillary. This is basement membrane, and this is the cell that form the wall of the DCT and collecting duct. Okay, it can be DCT, it can be collecting duct. So let's look at what happens when ADH arrive to these two tubes. So it will bind to, ADH will bind to the receptor. Okay, this is receptor for ADH. And then you already learned cell signaling, right? So this is a revision about cell signaling. So the ADH is the primary messenger, right? Primary messenger. So you already learned primary messenger. Okay, and because ADH is protein, ADH is a protein, it cannot pass through the membrane. It cannot pass through the membrane. So once it binds to the receptor, then the cell will have to activate a secondary messenger to be produced, right? Okay. So what happened that, so after ADH binds to receptor, it will stimulate cyclic AMP to be produced. CAMP stands for cyclic AMP. So this is a secondary messenger. So this secondary messenger will then activate a series of enzyme cascading reaction. So you can see here, this is a series of enzyme cascading reaction. Okay, after cyclic AMP is being produced. So this is CAMP. Yeah. Okay, then after that, activate a series of reaction here. So finally, what happened is that in the last step of this series of reaction, phosphorylase enzyme, ACE, right, is an enzyme. Phosphorylase enzyme will be produced. So this enzyme will catalyze the phosphorylation of aquaporins. Okay, so what is aquaporins? Aqua stands for what? Aqua? Water, right? Porin? Porin is channel protein. Okay, porin is pore. Pore means channel protein, because your channel protein that it forms a pore, water permeable pore in the membrane. Right? Let's say this is membrane, right? Okay. So these two are the protein that forms a little pore through it. So this is a channel protein. So this channel protein for water only, because aquaporin. So the enzyme phosphorylase will phosphorylate the aquaporins, and that will actually cause the vesicle, this is a vesicle containing aquaporin, will move towards the luminal cell surface membrane. Luminal stands for lumen. It's an adjective. It describes that this cell surface membrane is located at the lumen of the cell. Okay, so the vesicle will move towards luminal membrane and fuses with it. Okay, so now the luminal membrane is permeable to water because there's aquaporin. Okay, so now the water, you can see that now the water will actually move out of the DCT and collecting duct. So move from the lumen through the membrane, through the DCT and collecting duct cell, and go into the tissue fluid, and from tissue fluid in the medulla goes into the blood. Okay, the blood either in peritubular capillary or vasa recta, depending whether it's DCT or collecting duct, correct? Are you following? Yes, huh? Now, the next thing is, if water moves in this direction, there's a water potential gradient, right? There's a water potential gradient, right? So what is driving water to move is, remember just now I said sodium chloride? Loop of Henle actually reabsorbs sodium chloride, causing the medulla got a lot of sodium chloride, reduces water potential. So it is the sodium chloride that creates the water potential gradient, therefore causing the water to be able to reabsorb back from here. Okay, so it's because of the sodium chloride. Now, other than sodium chloride, what actually also reduces the water potential here is urea. Animation, the sound animation. Okay, urea. Now, when ADH arrives to the cells, right, the cell also becomes permeable to urea. So that means more urea will be reabsorbed. Okay, more urea will be reabsorbed into the tissue fluid. Okay, so I'm going to write here urea. So more urea will also be reabsorbed back into the tissue fluid. And because urea is soluble, right? So it's also a solute. So when the solute increases, which is urea, water potential, what happens? Drop, isn't it? Water potential will drop. Okay, so that will create a water potential gradient. Water potential gradient is being created. So that water potential gradient will cause the water to move in. Why is Chris coming in? Water to move in. Okay. Where does the sound come from? There or here? Okay. So water moves down water potential gradient by osmosis. Every time water passes through the membrane is called osmosis, right? Okay. So water moves down water potential gradient by osmosis to the concentrated tissue fluid in medulla, which is created by loop of Henle, as well as increase in urea permeability of the collecting duct. So that means sodium chloride and urea both will cause the water to move out. Okay. And then it goes into the bloodstream. Right, so last part is urine. So now because you remove a lot of water, urine becomes concentrated and lower in volume. So this picture shows you an aquaporine, where water can diffuse across the aquaporine. Okay. So now we know that ADH will cause DCT and collecting duct to increase in water permeability by adding the vesicle containing aquaporine to the luminal membrane, and also to increase urea permeability in order to reduce water potential medulla. Well, urea will reduce this one to increase water reabsorption. So therefore, the response is there will be increase in blood water content because we take the water back into the blood. So by increasing water reabsorbed. So this is the entire stimulus response model that you want to use to explain this part. Okay, so if you're familiar with stimulus response model, you can use this one to explain. It becomes very crystal clear. Okay, so now here, when our blood is too dilute, we will not secrete ADH. So ADH level is low. So you can see that both the DCT, which is distal convoluted tubule, and collecting duct, they are impermeable to water. So here the water cannot come out. So water permeability becomes low, right? Okay, so water permeability will decrease. So therefore, the water reabsorbed will become lower. So because of that, the urine has got a lot of water, it becomes dilute urine. Okay, so this is when you drink a lot of water lah. But when you don't drink water, your blood will be too concentrated. Let's say the whole day you didn't drink water, or you exercise a lot, you sweat a lot, you didn't drink water. So ADH will be secreted, the level will be very high in our body. So now you can see in the diagram, both the DCT and collecting duct becomes highly permeable to water. Okay, and then the tissue fluid in medulla, right? You have got the increasing sodium chloride concentration and urea concentration as you go deeper and deeper into medulla. The concentration becomes higher and higher. So that helps to actually drive the water to be reabsorbed back to our body, because solute will reduce the water potential, creating water potential gradient. So water will be reabsorbed. Okay, so water permeability will increase to both the tubes. Therefore, it will also increase water being reabsorbed. And therefore, what happened to your urine? Highly concentrated, right? Okay, so urine becomes highly concentrated. Okay, so now let's look at this graph. This graph shows you concentration of fluid versus different location of the nephron. So you need to be able to explain for PCT, DCT, and collecting duct, which are in your syllabus. DCT is only for osmoregulation. Okay. Now, can you tell me why is it the concentration of the fluid in the PCT remains the same even though selective reabsorption occur? Remember, I teach you before, you need to know that the concentration of a solution, okay, it depends on how much solute you have and how much solvent you have. When the concentration increases, what has changes? When the concentration decreases, what change? Solute change, solvent change, or only one of them? Change means does it increase or decrease? So now here, the concentration did not change at all. But you know selective reabsorption occur, right? That means what happens to what happens to PCT in PCT? Okay, let me call Xin Ding. Xin Ding like staring blur blur. What is the solute that's reabsorbed? Same as the amount of solvent reabsorbed. Which one? Huh? Yeah, solute and water both reabsorb, right? It must be the same amount, isn't it? So out of all the solute and water reabsorb, which one has got the same amount? Which one? KE? I thought just now I list up to you already, what? Ong Shumin? No. Huh? Correct. Water Na plus. Na plus how many percent? 80%. That's why I asked you to memorize, isn't it? I asked you to memorize just now. Water how many percent? 80%. So because sodium ion is a solute, we reabsorb 80%. Water is a solvent, we reabsorb 80%. So if you reabsorb the same amount, is there a change in the concentration? No change. No change. So how to explain this one? You need to memorize this to explain. Okay. Now, next part is, let's see, collecting duct. Collecting duct when there is ADH and collecting duct when there's no ADH. So when there's ADH, the concentration increases very fast, right? So why does the concentration increase so fast? Megan? Huh? Yeah, the collecting duct. Yes, very good, because here water reabsorb, right? Water reabsorb. Okay, so water is reabsorbed. So therefore, your concentration will drop because you remove the solvent, right? Your solvent, which is water, drop. Water drop. So therefore, the concentration will becomes increased. It's more concentrated. Okay. Now, when there's no ADH, water is not reabsorbed, right? So the concentration remains the same, isn't it? Because you didn't reabsorb water and urea. So that you need to explain. Now, this one is showing you the flow rate. That means how much volume move in per unit time. Okay, so you can see that the volume of the renal fluid becomes lesser and lesser as it flows down. It becomes slower and slower because we remove more and more water already, right? And also as it flows further down the tube, the resistance will slow down the movement. So both the resistance and also the reabsorption will reduce the volume that can pass through per unit time. So it becomes less and less volume per minute that can flow. Okay. So the next one is, this is interesting. Now you see, this is the concentration of these solutes in 4 different locations of the nephron. First thing, you can see that glucose goes all the way down, right? So why is it the glucose concentration goes all the way down? Shannon? Oh, I know. Why does the concentration go all the way down? Faster. How many percent glucose reabsorb? Shannon? Huh? How many percent of glucose reabsorb? Do you want the glucose or you don't want glucose? You want. So how many percent you want to reabsorb? Huh? No, 100%. All the useful substances are 100%, isn't it? So 100% of glucose reabsorbed. That's why you can see the concentration drop all the way until it can go all the way to 0, okay? 100% of glucose reabsorbed. That's why glucose concentration drop all the way down. Okay, 100% reabsorbed. Okay, so now look at the collecting duct. Can you see sodium ion concentration, potassium ion, and urea? All these 3 increases. Okay, where you? Yeah. So this one urea concentration increase, this one K plus increase, Na plus increase. So why does the concentration increases? Why does the concentration increases? Any movement of K plus and Na plus in collecting duct? No, right? Okay. So when you talk about concentration, always talk about the solute and the solvent. My solute, I didn't reabsorb. Right? Sodium and potassium, I didn't reabsorb, right? Water got reabsorbed or not? Yes, my water is reabsorbed. So because of that, my overall concentration will increase. I remove water already, right? Okay, so therefore concentration will increase. So this one will increase. Okay. Now you see the urea, if you want to go more detailed than that, I think it's good to know this. You see the urea, it increases but slower than Na plus and K plus, right? Because you know that urea also will be reabsorbed, isn't it? In collecting duct. Whereas K plus and Na plus are not reabsorbed. So therefore, when you reabsorb solute, okay, this is reabsorb, urea reabsorb. Reabsorb, but water reabsorb. More water. So there are more water reabsorbed. More water absorbed than urea reabsorbed. Overall, urea concentration will still decrease, right? Because you reabsorb water. But it will not decrease so much. Oh, sorry, you decrease more. You decrease more because you also reabsorb urea. You take away the water, but you also take away the urea. So the concentration will drop more, right? Okay. So the concentration here, it will not increase so much because we also reabsorb urea compared to sodium and potassium. Okay. So this is very important where you need to talk about the concentration. Okay. So this is where you need to write an answer. Just now I mentioned all the answers already. Okay. Right. So recap the function of nephron part by part. The first one is Bowman's capsule is ultrafiltration. PCT is selective reabsorption. Loop of Henle is to conserve water. DCT is to find control of blood composition. And last one, collecting duct, is osmoregulation. Okay. For this part here is how the tubes will look like under the microscope. Okay. So we know that nephron stretches from cortex to medulla. So in the cortex, for all the tubes that you can see, it can be Bowman's capsule, PCT, and DCT, and a little bit of the collecting duct. Medulla, all the tubes that you can see are the loop of Henle. You see loop of Henle, you've got a thicker part and thinner part, okay? And then the collecting duct. And of course, all the associated blood vessels, right? The peritubular capillary and the vasa recta. So let's look at a closer view. So in the cortex, you'll be able to see the Bowman's capsule and glomerulus, and then those cells with microvilli. So this structure is the PCT. Those without microvilli, the border between the cell and the lumen is very clear one. This is the DCT. Then the tube where the cell surface membrane separating the cell are clearly visible. This is the collecting duct, because all the duct in a body, right? The membrane separating the cells are clearly visible one. Okay, and then of course the smaller tiny tubes here at the sides are all the blood vessel. So in this case here, peritubular capillary. So now you'll be able to know how to identify glomerulus, Bowman's capsule, PCT, and DCT. Okay, so PCT and DCT. This one got the shadowy structure, which is brush border or the microvilli. So this PCT. This one is without microvilli because clear border between lumen and the cell. So this is DCT. Okay, so this is in the medulla. All the tubes are arranged parallel. So when you see all of them are transverse section. So let's look at the tube. So we have got, this is collecting duct. Okay, this is collecting duct. This is collecting duct. Then this, the rest of the tube, this is thick loop of Henle. This is thin limb of loop of Henle. Okay, and then those tubes where the lumen contain blood, they are the blood vessels. The lumen is empty, is the nephron. Okay, so this diagram is to show you, for you to learn out the structure. This is a longitudinal section. Okay, longitudinal section, this transverse section of the tube. Okay, so these are essay, question and answer. So you read it at your own time to go through what are the important points. So same as for this one. Okay. So can you see they underline the word posterior? So if you don't write posterior, you don't get mark for pituitary gland. Okay. So there's also, these are some of the essay questions. All right. For this part here. So that's all for today. All right. So you have a good weekend, all of you. All right. Bye-bye.
Thank you, teacher.
Thank you, teacher.
Okay.